Muscle contraction is probably the longest step-by-step process in all of A-level biology. In the myofibrils of skeletal muscle fibre cells, there are two key proteins, actin and myosin. Actin has binding sites where myosin heads can bind, but when a muscle is relaxed, those binding sites are blocked by another protein called tropomyosin. Each myosin head has an ADP and an inorganic phosphate attached. When acetylcholine is released at the neuromuscular junctions, the sarcolemma depolarizes. This depolarization spreads through the muscle fibre cell via the T-tubules and reaches the sarcoplasmic reticulum. The sarcoplasmic reticulum releases calcium ions. Calcium ions diffuse into the myofibrils and bind in a way that causes tropomyosin to change shape, moving it away from the binding sites on actin. OCR and Edexcel students, watch out that you know the role of troponin in this part. Now the myosin heads can bind to actin, and actin and myosin cross bridges form. The myosin head then changes shape in what's known as the power stroke. ADP and inorganic phosphate are released, and the actin filament is pulled over the myosin filament towards the centre of the sarcomere. At this point, a new ATP molecule binds to each myosin head. This causes the actin and myosin cross bridges to break, so the myosin detaches from actin. The ATP is then hydrolyzed to ADP and inorganic phosphate by an enzyme called ATP hydrolase that was activated by the calcium ions. ATP hydrolysis releases energy, which allows the myosin head to reset back to its original position, ready to bind to the next binding site further along the actin filament. The myosin head attaches again, another power stroke occurs, and the cycle repeats. As this happens across many sarcomeres, the muscle shortens. When stimulation stops, calcium ions are actively transported back into the sarcoplasmic reticulum. As the concentration of calcium ions in the myofibrils decreases, tropomyosin moves back to block the binding sites on actin. Actin and myosin bridges can no longer form, the actin filaments slide back to where they started, and the muscle relaxes. It's not simple, but this is some really cool biology in action, and hopefully this video helps your understanding.